Good evening, students. So far in the chapter of human reproduction, we have completed male as well as female reproductive system, and we have also studied the gametogenesis process in males, that is what we call it as spermatogenesis, that is process of formation of male gamete, that is the sperm cell. Now in this segment we are going to discuss about the process of formation of ovum or egg in the female body, and the process is what we call it as oogenesis. So today our task is to complete oogenesis. So straightforward, oogenesis means the process of formation of egg or ovum in the female body. So oogenesis is process of formation of egg or ovum in the female body. Talking about the location of oogenesis, the location of oogenesis is specifically the ovaries of female reproductive system. Location is within the ovaries. Now we have also discussed spermatogenesis, and the spermatogenesis was a continuous process. Oogenesis is a kind of discontinuous process. This statement is very important. It is a discontinuous process. Now when I am saying it is a discontinuous process, now what is the meaning of discontinuous process? Discontinuous process means this process is not a complete process, right? This process is not a complete process. There are various stages of this process. These stages takes place, or we can also say that oogenesis starts when the female embryo, when the female child is within the womb or the uterus of the mother cell or the mother body. So it is a discontinuous process, because certain stages of oogenesis takes place during the embryonic development, certain stages takes place at the birth, certain stages takes place at the puberty age. So like there are various segments of stages involved. So that's why it is a discontinuous process. Now before going into the details of oogenesis, let us start with some numerical fact regarding oogenesis in terms of follicles. We know that there are follicular cells, specifically the granulosa cells. These cells are responsible for the formation of follicles. For the formation of follicles like primary follicle, secondary follicle, tertiary follicle. Tertiary follicle will undergo certain changes to form a Graafian follicle, and that is what we call it as a mature follicle. And this Graafian follicle or mature follicle consists of secondary oocyte. Right? Now, during the first month of embryonic life, the number of primary follicles that are present are around 1700. Around 1700. Now what is going to take place? These primary follicles will multiply, they will proliferate, primary follicles will multiply, they will proliferate and increases its number. So during the first month of embryonic life, the total number of the primary follicles that are there in the female body is 1700, that is within the ovaries. And let us say during the second month of embryonic development, these follicles changes to 6 lakh. You can see the great increase in the number of primary follicles. Again, proliferation, multiplication, growth of the primary follicles will take place. Let us say during the 5th month of the embryonic development or the embryonic life, the number of the primary follicle reaches to 7 million. So 1700 during the first month of embryonic life, 6 lakhs during the second month of embryonic development of life, 7 millions during the fifth month of embryonic development. Now what happens next? There is a stage which we call it as follicular atresia. Now the follicular atresia will start, that means decrease or shrinkage in the number of the follicles will take place. So the stage is called as follicular atresia. What is follicular atresia? Follicular atresia is basically decrease in the number of primary follicles. So up till fifth month during the embryonic development, the total number of primary follicles was 7 million. At birth, this number will decrease to 2 million. So at birth, the total number of follicles that are present are approximately 2 million. This process continues to take place till puberty and then it stops. So during the puberty stage, the number of the primary follicles are in several thousands only. That is around 60,000 to 80,000 in number. Now what is going to happen next? After puberty, no new primary follicle will develop. No new primary follicle will develop. If let us assume that out of 500 total primary follicles from 60 to 80,000, we know only one primary follicle will go up to the maturation stage to form something, that is what we call it as Graafian follicle. So from, it is just an assumption, 500, only one primary follicle will mature. And what is it going to form? It is going to form Graafian follicle, and this Graafian follicle is also called as mature follicle. And we have already studied the structure of Graafian follicle in detail. The fluid filled cavity, that is what we call it as antrum, and the secondary oocyte, that is well protected within the discus proligerous cells of Graafian follicle as well as granulosa cells. Now what is the next? The oogenesis process. Basically the three main steps of oogenesis are multiplication, growth as well as maturation phase. So three phases. Of oogenesis. Before this, oogenesis is a process of formation of the egg or ovum in the female body, basically in the ovaries. And the development of ovaries start from or between 11th to 12th week of pregnancy period. The development of ovaries begin, but it is completed till the 11th to 12th week of pregnancy period. The ovaries are very well developed within the female reproductive system, mainly in the abdominal cavity. So three phases of oogenesis are multiplication phase, second phase is the growth phase, and the third phase is called as multiplication, growth as well as maturation phase. Let us start the three phases of oogenesis one by one in detail. So first step, that is multiplication phase. Now this segment will be reserved for theory, and in this area the diagrams pertaining to oogenesis will be drawn. So the first phase is multiplication phase. We know that the germ cells are present, that is the germinal cells, or you can also say the primordial cells. These primordial cells will divide mitotically undergo successive mitotic division to form something that is called as oogonia. Right? So you can write that primordial or follicular cells divide mitotically to form oogonia, and this oogonia undergo one more mitotic division to form something that is called as primary oocyte. Right? So this is the first step, that is multiplication phase. So what happens? Let us say this is the primordial cell, that is the germinal epithelial cell or follicular cells. It is primordial cell, the ploidy number is 2n, it's divide rapidly, undergo mitotic division to form something that is what we call it as oogonia. Let us say this is the oogonias which are developed. If we are saying it as 1, so what we will say? Oogonia. Okay? So this is again a diploid cell that is called as oogonium. Oogonium again undergo mitotic division to form something that is called as primary oocyte. If you remember, in case of spermatogenesis, primary spermatocyte was formed. In case of oogenesis, primary oocyte will develop. Now the next phase is growth phase, and we all know what happens in the growth phase is just the accumulation of the nourishment, that's it. So the next phase is growth phase. What are the peculiar characteristics of the growth phase? Basically, the yolk content develops, that is the fat accumulation takes place. So fats and proteins are in yolk. Cytoplasm is rich with, cytoplasm contains ATP, enzymes, DNA, RNA. Other cellular organelles like mitochondria as well as Golgi body and endoplasmic reticulum also present or also develop within the primary oocyte. So just for conclusion, in the growth phase, primary oocyte undergo only growth by accumulating all this nourishment, all this nutritive material within itself, and the size of primary oocyte increases. So up till now, that is in the multiplication phase as well as growth phase, the divisions were purely mitotic divisions. The divisions were purely mitotic division. Now what is going to take place at the next maturation phase? In that, meiosis will take place in two subsequent steps, that is meiosis 1 and meiosis 2. So this primary oocyte will now enter into first meiotic division, and because of the first meiotic division, two cells are formed. One cell is a bigger cell and one cell is a smaller cell. The bigger cell with its nucleus is called as secondary oocyte, and the ploidy level is haploid. It is haploid. Why it is haploid? Because it is undergoing the meiotic cell division, and that is what we also know it as a reductional cell division. That is from 2n it will lead to n. So this big cell is called as secondary oocyte, and the smaller cell, which is again the ploidy n, is called as first polar body. So this structure is called as first polar body. Now why this one smaller cell and one bigger cell? The reason is very simple. Why simple reason is, we know that from these follicles only one primary follicle is going to mature, to produce one mature Graafian or mature follicle. That is what we know from previous discussion. So only one oocyte is going to develop from one mature Graafian follicle. So that's why unequal cytoplasmic division of the primary oocyte takes place. So what is the statement? Statement is unequal. It is unequal cytoplasmic division. Unequal cytoplasmic division occurs in such a manner that one cell receives maximum of the cytoplasm, and the cell that is receiving the maximum cytoplasm is called as secondary oocyte, and the cell which is receiving the less amount of cytoplasm is called as first polar body. So unequal cytoplasmic division takes place of primary spermatocytes. This is meiosis 1. Maturation phase is not yet completed. Secondary oocyte will undergo second meiotic division, right? Second meiotic division, that is meiosis 2 will result, and we know meiosis 2 is equivalent to mitosis, that is equational cell division, because we can go from 2n to n. We cannot go from n to n by 2. That is not a possible case. The Ploidy number cannot be n by 2. If it is n in meiosis 2, it will always remain n, because meiosis 2 is equivalent to mitosis. Secondary oocyte will result in the formation of ovum or egg, or you can also say ooted. And again, here also one smaller cell and one bigger cell will develop. So this bigger cell is ovum, egg or ooted, and the smaller cell is the second polar body. So this is again n, this is again n. So this is second polar body. Now this polar body has no significant role in the oogenesis process. So gradually, slowly and gradually they degenerate from the female body, that is from female reproductive system. One important thing that we need to take care, or we need to know as far as competitive exams are concerned, in the growth phase. Growth phase is basically the accumulation of all the nourishment of the nutrient in the egg cell, and the process on which the growth cell is dependent or growth phase is dependent is called as vitellogenesis. So the process is called as growth is totally dependent on vitellogenesis. Vitellogenesis. Vitellogenesis is basically the accumulation of the yolk content in the egg cell, right? In case of human beings, the yolk content is approximately 200 times. That is the yolk content. So what is going to happen is this oogonia size increases to primary oocyte. So 200 times increases observed in case of human females. In case of hen, it is again 200 times more yolk content is present. In case of frog, it is 2000 times, and in case of Drosophila, the insects, it is around 90,000 times more yolk content. Now, why this variation in the yolk content? This variation is because of the developmental stages that a particular organism is showing. Hen, frog and Drosophila, basically they are egg-laying organisms or egg-laying animals. Human females do not lay any egg. The entire embryo will develop into fetus, and fetus will give rise to the young ones. Hen, egg, frog as well as Drosophila, basically they are egg-laying animals. So they lay egg. Again, when they lay egg, the development process is not yet stopped. If you see in case of frog, larva stages are released from the egg, that is the tadpole structure, and then through the metamorphosis structure, the larva changes into adulthood stage of the frog. So for that developmental processes, nourishment is required, and the nourishment is obtained from the yolk content of Now coming back to this point, it is a discontinuous process. Now let us understand why this is a discontinuous process. We have seen this follicular phases, that is during the first month it is 1700, second month it is 6 lakhs, fifth month 7 million, then at birth 2 million because of the follicular actuation process. This process is again a genetically defined systematic process. Puberty 60 to 80,000 and only one will give rise to primary follicle, that is the Graafian follicle will develop. So up till here, that is the follicular stages takes place during the embryonic life only. That's it. Up till here. At birth, all these phases, that is multiplication phase and the growth phase, takes place till the birth. Then, when the primary oocyte is converted into secondary oocyte, this secondary oocyte development takes place till puberty. Now, at puberty, this process is arrested. That means this process stops, and this process stops during the diplotin phase of the prophase 1 of meiosis. So, what is that phase called as? Diplotin stage of the prophase 1 of meiosis. So, this is arrested at diplotin stage of prophase 1 of meiosis, that is meiosis 1. So this is up till now meiosis 1 has happened and this process arrested. Ovulation takes place in this stage only. Now secondary oocyte is developed. At this stage ovulation can take place, but when secondary oocyte develops into oocyte, that means the meiosis 2 will be triggered only and only when insemination takes place. So in order to get triggered meiosis 2, insemination takes place. When insemination takes place, only and only then meiosis 2 will start. Before that, secondary oocyte will remain for a very long period of time, for years, within the female reproductive tract, that is in the ovaries only. When the female is sexually stimulated, that is when the sperm enters into the vagina of the female body, they travel to the fallopian tubes, insemination takes place, and then secondary oocyte undergo meiosis 2 to form the ovum or the second polar body. So this is all about the mechanism of oogenesis. In the next part, we will discuss about the structure of human egg cell.